Hi students, today we're going to see a new topic which is the embryo development. Hope you enjoy it. Now, we're going to start by answering a question. Where does an embryo come from? And you need to remember that the embryo, which is a very important part of sexual reproduction, comes from the union of two haploid cells that we call as gametes. The female gamete will be the ovule, an haploid cell from the females, and the male gamete, an haploid cell as well, but from males will be the sperm. Once these two haploid cells join, they are going to form a single cell, a very big one, called the zygote. We say that is the first cell produced after fertilization, which is the process in which the gametes join. Now, this is a real image of a zygote, so you can figure how does it look in real life. It is surrounded by a protective layer of cells that avoid other spermatozoids or sperms to join in. Just one must join into the ovule in order to form the zygote. And then inside, the two nuclei are going to fuse to have a diploid cell. Okay, moving forward. Then, the stages for embryo development are divided according to the number of cells and the structures that we have inside this zygote or this further structures. First, you need to remember that the divisions of the zygote, they're not longer called as mitotic division. It's a special type of division called as the cleavage. Now, why is it special? Because in cleavage, we don't have the time for the cell to regrow again. In this case, the cytoplasm will be reduced each time more and more until we reach a normal size cell. Remember that the zygote is very big, right? So when the cleavage is produced, the next or the new cells are going to have less cytoplasm each time until they reach a normal size of a cell. Now, cleavage usually produces two cells at the beginning. Remember that we come from a very big cell, just one. Then with the cleavage, we produce two cells at the first stage. Then we have four stage, then eight, and we go on and on and on until we reach the next stage of the embryo development known as the morula. But first, there's a real image of the cleavage. This is how the zygote divides in the first two cells. Then, as I was saying, the next stage is the morula. How do we define the morula? It's a solid ball of cells that come from the cleavage. Now, some of these cells are going to migrate to the external part of the morula, leaving a hollow space inside. Because of this difference in the structure, we have a new name for this, and this is the blastula or the blastocyst, which is this structure. Now, remember that the key word or the key structure, the main difference of the blastocyst of the blastula is the hollow space inside. Now, this is the real image of a blastula or a blastocyst, right? Okay, so moving forward to the next stage. The next stage, this is just a summary for you. We have a zygote, one single cell with cleavage or the divisions. The cell is going to divide first in two, then four, eight, and we go on until we have a solid ball of cells called the morula first, and then the blastula. How do we know that this is the blastula? If we divided this, we will see a hollow space. Now the next stage, when some of these cells, probably this section, they're going to form this structure known as the archenteron, which will eventually form the gut cavity or the organs inside the embryo or the organism. Now, this structure, this special structure, it has a special pore at the beginning where everything started, and this is called the blastopore, which eventually will produce the anus of the organism. And this gastrula. Another characteristic of the gastrula is that it has three main layers clearly identified, and you already know them, which is the endoderm inside, the ectoderm outside, and in the middle, the mesoderm, right? So here you have a picture or an image of a gastrula, a real gastrula. Here we can see clearly the three layers. The ectoderm, this will be the mesoderm, and this will be the endoderm. Remember that this is the archenteron, This is the blastopore that will produce the anus of the organism, and these will produce the gut. Okay, so moving forward. Remember that the gastrula has three main tissues, and these three tissues are the endo, the ecto, and the mesoderm. They produce different parts in the adult or the grown-up organism, the already mature organism. The ectoderm, which is the external part, will produce the skin, the nervous system, some sensory organs. The mesoderm, which is in the middle, will produce the parts of the connective tissue like the bones, the blood, the muscles, and the endoderm, that is the most internal, will produce the internal organs like the gut, the liver, the pancreas. So, how can these tissues turn into different organs? With a process known as organogenesis, which is a way in which the tissues differentiate or produce new organs, right? You need to remember that the embryo has like pure cells, very powerful cells that we call as stem cells. They don't have a particular function. And once they got their function, they differentiate into special organs. This process is called organogenesis. Now, another way in which these cells can differentiate and produce these special tissues is because of a process known as induction. Now, what is induction? Induction will be when some cells affect the surrounding cells by a type of communication that you know as direct or paracrine communication, if you remember those concepts. So what does this image represent? Cells type B will induce the production of new cells in cells A. The new cells are cells type B. And cells type B will induce these two to produce new cells like D and E. So you can see it's like some cells deciding the fate of the surrounding cells. Now, another way in which these tissues could differentiate is by homeobox genes. What are those genes? In the genome of an organism, these homeobox genes will control the development of very big areas of the body. They are generally located in the same order as the parts of the body are located. So if you can see here, this gene will direct the production or the development of the mouth of this organism. This gene will produce the forehead. This gene will produce the back part of the head, and we go on and on and on. So they are very important genes that direct very big parts of the organism. Finally, you need to understand that the embryo is going to be surrounded when it is more development than the rest of the or the previous stages. The embryo is going to have four external membranes that they have special functions. So the first membrane is the chorion. The chorion will lead to the part of the placenta that the embryo produces, because the placenta is formed by two sources. One is provided by the mother, and the other is provided by the fetus. The next embryonic layer is the amnion. The amnion, it's like a bag or a sac that is full of liquid that provides a watery environment for the embryo. And also, that's why we call the egg or the ovum, in the case of the humans, an amniotic egg, because it's going to be a watery environment for the embryo. The next layer is the allantois. The allantois and the yolk sac, they have the special function of producing the umbilical cord, which will eventually connect with the mother in order to diffuse or exchange nutrient and oxygen. So I hope that this video is useful for you. I have an activity that will help you to summarize everything, and is this type of table. You can pause the video and try to figure the concepts according to the definition. And if you can't remember these concepts, then, well, I will have the definition for you. So the first cell that is formed after fertilization is known as the zygote. The type of division that the zygote undergo to produce more cells is the cleavage. A solid ball of cells that come from the zygote is the morula. Then the hollow ball of cells is the blastula. Once we have three main tissues clearly identified with an invagination or a group of cells that forms like the archenteron on the gut, we have the gastrula. All the genes that control big parts of the development of the body, the amniobox genes. The induction, the process in which a gene affects the the cells affect adjacent cells by a paracrine or direct communication, and they control their fate. And finally, four main layers that form the placenta, the fluid, and the umbilical cord will be the external embryonic membranes. I hope you enjoyed this video. I hope it's useful for you, and we'll see you yourselves in the next video. Bye!